All right, so the next major section that we're going to be talking about is going to be the macromolecules of life, or the major molecules of life. And if you remember, we sort of talked about this a little bit earlier when we talked about common characteristics of life. We talked about how there are certain biological macromolecules that are found in all living things. So we're going to just sort of start at the beginning. We're going to intro, we're going to talk about all these major molecules of life. Then we're going to break them down into their different categories. So just as a background, it was once thought that all of the molecules of cells must contain some sort of vital force that differentiated them from the molecules of non-living things. And so because of this, all of chemistry was broken into the divisions of organic, or the chemistry of living things, and inorganic, the chemistry of non-living things. So it was considered that anything that was of a living source was organic, anything non-living was inorganic. We no longer recognize this idea of vital force in order to differentiate two molecules, but we have kept this division for our own understanding. We now identify things as organic or inorganic molecules, but our definition has changed slightly. So instead of, what do you think of when you think of the term organic? A lot of individuals, or a lot of people think of natural, fresh, healthy even. Organic would be of natural origin. And all of these things are not wrong, but that comes from more of a modern or a current understanding of the term organic. Organic actually means, in its scientific definition, it means molecules that contain both hydrogen and carbon atoms. And that's it. That's as simple as the definition gets. Anything that contains both carbon and hydrogen is considered an organic molecule. So for instance, glucose, which is C6H12O6, has 6 carbon atoms, 12 hydrogen atoms, and 6 oxygen atoms. So glucose is an organic molecule, contains both carbon and hydrogen. But what about water? Is water organic or inorganic? So water's molecular formula is H2O, meaning there are 2 hydrogen atoms, 1 oxygen atom. So my question is water organic or inorganic? Same with carbon dioxide. Is carbon dioxide CO2? So 1 carbon, 2 oxygen. Is that organic or inorganic? What about oxygen? Is oxygen organic or inorganic? So just some things to think about. Let me know. What do you think about whether those molecules are organic or inorganic? But we're going to move on to studying the classification of organic molecules. Specifically, we're going to break them down into one of 4 categories. We're going to be talking about carbohydrates, nucleic acids, proteins, and lipids. So these biological macromolecules are going to be giant molecules. The reason we say they are giant molecules is because in comparison to things like carbon dioxide or water or even glucose, these are going to be very, very large molecules. Almost all of them are going to be made up of long chains. So, for instance, carbohydrates, complex carbohydrates, like starch, is going to be a polymer, or a long chain of a polymer made of many repeating units called monomers. So starch, for example, is a polymer made out of glucose. Glucose is the monomer, that's the single unit. A whole bunch of glucose molecules all strung together in a great big long chain is called a polymer. Poly meaning many, mono meaning one. So a polymer is made up of many, many, many monomers, single monomer units. Now, the interesting thing is despite the huge amount of diversity in these macromolecules, there's a huge range of even the number of carbohydrates. There's a huge range in the number and size and types of carbohydrates. There's a huge range in proteins. But even just proteins are so very different from carbohydrates. Despite all of the diversity within these major macromolecules, we're going to see some very similar things that they all sort of have in common. And one of those things that they have in common is that, one, they're all polymers made up of monomers, but that these polymers are assembled in the same way. We're going to use the exact same reaction to string a whole bunch of glucose molecules together to make a starch that we would use to string a whole bunch of amino acids together to make a protein. protein. So it's going to be the same type of reaction. So we're going to learn what those basic reactions are. The first one is going to be a dehydration reaction. The reason we call it a dehydration reaction is because it is a chemical reaction in which those subunits are joined together by the formation of a covalent bond. And while we are forming this covalent bond, water is going to be produced and released during this reaction. This is going to be used to connect monomers together to make polymers. So think of stringing beads together on a chain, or linking cars together to make a train, like a bunch of cars in a choo-choo train. Sometimes I'm not sure how clear I can make myself on this video. But yes, so all the cars of a train strung together. We're going to connect those up through a dehydration reaction. So a great example of this would be the formation of starch from glucose monomers. We're going to take glucose monomers, we're going to undergo a dehydration reaction, kick off a water molecule, and glue them all together. Then, if we want to break them down, we can take these macromolecules and we can break them down using a very similar converse process. This would be known as a hydrolysis reaction. Hydro meaning water, lysis to split apart. We are going to actually shoot water molecules at that covalent bond and break them up. So a dehydration reaction is we string them together and release water. A hydrolysis reaction is we add water to break them up. You can kind of think of it the same way we dissolve anything. If you had a cracker, saltine cracker, and you stuck it in a glass of water and stirred it around, that water would break apart all of those bonds. It would break apart that starch, it would break apart those molecules, and break apart that cracker. So we're going to use water to break these covalent bonds. We're going to use these to break polymers down into monomers. So we're going to take a polymer, break it into monomers. A great example of this would be the digestion of starch into glucose monomers. In your mouth, you have a molecule known as amylase. It is an enzyme that turns starch into glucose. And so this is a lot of words. I know this is a lot of words. It's not a whole lot to see, but I wanted you to have this written down so that way you can have it in front of you as we go through these next couple of slides. So here's an example of how we synthesize biomolecules. You see that term synthesis? That means to bring together or to build up. And so what we're going to do here, we have our polymer. We have a bunch of, well, in this case, only two, but we have two monomers strung together already. So this is going to be our big polymer, and we want to add another car to this train. What we're going to find is over here, we have a hydrogen atom, and on this side we have a hydroxide atom. When we bring this monomer to join the polymer, this hydrogen is going to join this hydroxide, and suddenly we're going to have two hydrogens and one oxygen. What does that equal? H2O. Two hydrogens, one oxygen, and we have water. So when these two go off together, we're going to have sort of an empty hole right here where these two atoms used to be. They're going to join together to form a covalent bond. This is what's known as a dehydration synthesis. We have a chain, we have a monomer subunit, we dehydrate them, kick off a water molecule, and we suddenly have a beautiful long chain of polymer. Conversely, to break down that polymer into monomers, we're going to undergo a degradation. Degradation is breaking it apart, degrading it, breaking it into its smaller components. To do this, we are going to add a water molecule. When we add a water molecule, the hydrogen ion is going to join the polymer chain on one side, the monomer chain on the other side is going to be joined up with the hydroxide ion, and now we're going to split. We have one chain made up of multiple monomers and one single monomer over here. And that's going to be our degradation. And whether we are talking about carbohydrates or lipids or proteins or nucleic acids, they are all going to be built up, synthesized in the same way. They're all going to be synthesized through a dehydration reaction, a dehydration synthesis. They're all going to be broken down through a hydrolysis reaction. So a couple things I want to say about synthesis and degradation. I do want to point out that these are non-spontaneous reactions. So I think my saltine in the water reaction is a little bit convoluted. Things are not going to just start breaking down automatically. Just because you add something to water, specifically starch, starch is a very stable molecule. It's not going to just break down right away in water. It's actually going to require the use of enzymes. Enzymes are molecules that speed up a chemical reaction. Given enough time, eventually someday starch will break down into its monomers, into its glucose monomers. But it takes a very, very long time, and it takes a great deal of energy to get that process started. An enzyme, on the other hand, is a molecule that can speed up that chemical reaction. Enzymes are unique. They are what are known as catalysts. They're not part of the chemical reaction. Starch goes in, glucose comes out. Enzymes aren't part of the chemical reaction, but they do speed up that chemical reaction. They won't be consumed by the reaction. They're not changed by the reaction. They are merely catalysts. They just sort of help that reaction along. And I believe we're going to talk in greater detail about enzymes at a later time, so I won't go into detail about it now, but keep that in mind. We're coming back around to enzymes functioning as catalysts. Here I have a video that is about the synthesis reaction. And unfortunately, it looks like while I am recording, I cannot play this video for you. So we're going to set this video off to the side, and this will be maybe an opportunity for you to come up with your own extra credit tutorial videos about synthesis and degradation reactions. All right, so we are going to, our next step is going to move into the different major molecules of life. So we'll talk about carbohydrates, proteins, lipids, and nucleic acids. And we're going to have separate videos for each one of those. So that was our overview of the major molecules of life, and I'll see you in a moment for our discussion on carbohydrates. All right.